Assalamualaikum and hi everyone. I am responsible to present topic 2.0 cell structure and function, subtopic cell membrane and organelle. Our learning account is to show the detailed structure of typical plant and animal cell and state the organelle presence in one. By using light microscopes, can just observe protoplasm. This means that we can only view nucleus and cytoplasm surrounded by a thin plasma membrane in a large polymer wall that contains the granule and often temporary small vesicles. The organelles are mostly too small to be seen. Here is mineralized animal cell as seen with a light microscope taken from human chick squamous epithelial cells. Show nucleus and thin cell membrane. Here is mineralized animal cell and plant cell as seen with a light microscope. Only seen cytoplasm, cell wall in plant cell, nucleus for both animal cell and plant cell, chloroplast in plant cell, large vacuole in plant cell, and plasma membrane for both animal cell and plant cell. Here is mineralized animal cell as seen with the electron microscopes. Show nucleus, rhizome, the Eukaryotic, mitochondria, Nicolson, cytoskeleton and surrounded its plasma membrane. Not found in animal cell, chloroplast, central vacuole organelles, cell wall, and plasmodesmata. In plant cell, there is a protective rigid cellulose cell wall surrounding the cell. Cell wall composed of cellulose, pectin, hemicellulose, and some protein which provide structural strength and protections. In plant cell, a large central vacuole filled with cell sets is present in mature plant cell. The vacuole is surrounded by a specialized membrane called tonoplast. The green organelle only seen in plant cell is called chloroplast. Chloroplast contain polypigment which carry out photosynthesis. Here is mineralized plant cell as seen with an electron microscope. Nucleus, Eukaryotic, mitochondria, cell wall, chloroplast, cytoskeleton, tonoplast, central vacuole, ribosome, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Found in plant cell, lysosome, centriole, and flagella. Adjacent plant cell wall are joined together by the middle lamella between them. The cell wall may contain a number of small holes called pits. These pits allow strands of cytoplasm called plasmodesmata to link different cells together. What is plasmodesmata? Plasmodesmata is fine cytoplasmic strands that connect the protoplast of adjacent plant cells by passing through their cell wall. Here is differences between plant and animal cell. First, for animal cell, no cell wall, surrounded only by plasma membrane. While in plant cell, surrounded by a plasma membrane and a cellulose cell wall. Shape for animal cell is irregular and can be altered. while in plant cell it has fixed and regular shapes. Animal cells do not have chloroplasts, while plant cells contain chloroplast to carry out photosynthesis. Animal cell has small vacuole, while plant cell has a large center vacuole that is surrounded by nucleus. In animal cell, cytoplasm, organelle, and nucleus are not pushed to the periphery, while plant cell, cytoplasm, organelle, and nucleus are pushed to the periphery due to the presence of large center vacuole. In animal cell, food is stored in glycogen, while in animal cell, food is stored in the form of starch granules. Animal cell has a pair of centrioles, while plant cell no centriole. Lastly, lysosome almost always present in animal cell, while in plant cell, lysosome not normally present except some species like Nicolson. Next subtopic, the structure and function, cell membrane, and organelle. So our learning outcome is to show the structure of plasma membrane based on Fluid Mosaic Model CL1. Here is the component of plasma membrane. First, phospholipid. Second, globular protein including integral protein, chloroplast. Third, carbohydrate which is made up component of glycoprotein and glycolipid. Lastly, cholesterol. Here is the component of plasma membrane with phospholipid bilayer, globular protein, integral proteins, carbohydrates, which is component for glycoprotein and glycolipids. The last one is cholesterol. For Fluid Mosaic Model structure, there are two components to study. First, major component, phospholipid and globular protein. The other component called carbohydrate and cholesterol. Cell membrane is a general term that refers to all the biological membrane found inside and enclosing the eukaryotic cell. All cells are covered by a thin cell membrane which separates the cell content from the extracellular environment. Eukaryotic cells have membrane-bounded organelles. The cell membrane surrounds organelles such as nucleus, mitochondria, and chloroplast. The plasma membrane and the organelle membrane have the same basic structure. In 1972, S.J. Seymour and J.L. Nicolson proposed the Fluid Mosaic Model. Cell membrane consisting of a bilayer of phospholipid with various protein molecules embedded and attached to it. This structure is selectively permeable, meaning that it allows the passage of some molecules, especially of water, but not of others. According to this model, a mosaic because the proteins are embedded in the phospholipid bilayer, while fluid because the protein and phospholipid are free to move.